Welcome to Pigeon River Farm, doing farming right. I'm Robert Brown, the owner of Pigeon River Farm. Thank you for viewing.
Okay, I guess we're going to get started. I think pretty much all of you have been to a meeting before. Keith is going to be presenting some different things, and Keith is from Zone Products, and I've worked with Zone for probably what?
Zone?
Yeah.
Zone Products? Since 1992.
So with that, we'll get Keith going. And throughout this whole presentation, if you have any questions, just run out and ask the question. It's always good to have involvement from the crowd. Keith?
If you do, like Brian said, if you got any questions, raise your hand. Don't be bashful. I'm going to go through about 3 different presentations here, because what Brian kind of threw at me, what he was wanting to try to cover, I kind of am taking parts of 3 different presentations that I kind of modified and put together. And this first one I'm going to go through, it's more informative for you in relationships to what most people call kelps, okay? And the reason why I want to kind of go through this and kind of going to start with this, because you have heard me talk about through the years, some of you have heard me talk about the differences in like the Zone foliar supplement and the kelp growth. Where I've made comments, they're both made out of kelps, but they're made out of 2 different types of kelps, and how so many different species of kelps. And that's what I'm going to kind of go through here. There's some pictures in here. Any of you that's kind of familiar with human health type aspects, there's going to be some names that's going to pop up in here. If you're very familiar with herbal type remedies and everything, you're going to see some names pop up in here that way. The term algae, typically when I throw out the term algae, most people think of a green pond, right? This pond that's stagnant, green on top, when we think of algae. And that term that we use in relationship to that is really a very broad term. Actually, all species of kelps falls underneath the category of algae, okay? Those are in, because algae is actually a very small plant type substance is what algae is, okay? So, the type that we talk about growing on a pond, that way. Another term that you probably heard used is seaweed, okay? Seaweed, kelp is going to be synonymous of each other. A lot of times we pick up these terms and it's from different fields of study, or if you're talking about someone that's studying the ocean compared to someone that's a plant pathologist, someone that studies plants, they tend to name things different, and us general population, we hear the different names, and sometimes we tend to think that those are 2 different objects, okay, when they're the same thing, okay? So, now, algae, kind of the definition you see there is an aquatic photosynthetic type organism. And I've got there in that, if you can read that, I hope that green shows up good. It is a group of extremely diverse, ranging from what they call giant kelps to microscopic type plants, that way. All that falls in the category of algae, okay? There is 5 divisions of algae. One of the divisions is Chromophyta. Also within that, I'm going to go through some of these scientific names, Balsophyceae, diatoms, and then you get your Chrysophyceae, golden algae, and then you get Xanthophyceae, which is your yellow-green algae. Probably some of you in natural health, you've heard people talk about blue-green algae as far as a supplement for a person to take. That falls in that category there, in that classification. And then you get your Phaeophyceae, which is your brown algae. Now, typically, as far as someone that's got animals that's feeding a kelp meal, it's going to fall under this brown algae category, okay? Now, one of the, I didn't put it on there in the previous slide. You get one of these, you notice there's a golden algae, there's a yellow, green, and there's a brown. I didn't get it put on there. This one here is actually part of the red algae classification, okay? The red algae division, there's two primary ones, Irish moss and dulse. Now here's two other terms that if you're very familiar with herbal type, human health type supplements, both of those, dulse is a very prominent thing that a lot of natural health people will use in relationship to the thyroid, okay, for supporting the thyroid. Irish moss is a substance, if you get more of a natural-based hand lotion or body lotion, most of them will have Irish moss in there, okay? Both of these is in the red category of kelps that way, okay? There's a picture of Irish moss, kind of looks like a red lettuce. It's not a real big plant. It grows in more shallow type waters, salt type waters. Your brown algae. Okay, now when we get into browns, anything that you typically think of, like I said, of a kelp meal or a feed kelp that you may use falls in this category, okay? Now if you notice here, this brown algae, there is 1,500 species of brown algae known to man at this time, as far as worldwide in the oceans, okay? That's why when I talk about it's a very, very diverse group, that whenever we throw out the term kelp or a kelp meal, a lot of times I'll ask, is that what type? And everybody says, well, it's kelp, it's seaweed. And it's like, well, there's thousands of species out there, and those species make a difference. And some of you heard me use the example. If someone from a city, if you just use the term cow, well, I have a cow. Well, you get someone from the farm, our next thing is, well, what type of cow? Is it a beef cow? Is it a dairy cow? You know, we can break down classifications, then you start, okay, you got a dairy cow. Was it a brown Swiss? Is it a Jersey? Is it a Holstein? Is it black or red Holstein? See what I'm saying? The same thing we get into when we just get into the brown kelp families. It's enormous at how this divides up. Now, a brown Swiss, or let's say a red Holstein and a red Angus, they're both red. They're two totally different. They're both bovine, right? But what they do and how they function is totally different, okay? The same thing goes when we talk about these different kelps. I'll throw up some of these different family names here. Bladderwrack is another one that you hear a lot talked about in human health aspects, with the thyroid especially. You can get your oriental food kelps. The Oriental people from Asia, in Eastern Asia, is very big on using kelps in their diet. They're very different from our Western European type societies, where a lot of our ancestry is from. We weren't as big on kelps. Now you get in Asia, maybe it's been in Asian type countries, Japan or China or Philippines or anything like that, they've got kelp at about every meal in some way. Okay, we just like, man, how can you cook kelp different ways? But they really keep that into their diet that way. Now, off the west coast of North America, there's a species that's called giant kelp, primarily from about San Diego, California, all the way up to the Bay of Alaska. There's a particular species, and that's the only place it's found in the world, is just off that west coast there. And it's huge. They even talk about it being like a forest under the water, these giant kelps that way. I think I got a couple pictures that way in here coming up. Now, you get the bottom one there is one that I want to talk about here, this sargassum, also known as gulfweed. There is, at this point, there's only 3 known fields of sargassum in the world. One is off the southeast coast of the United States. Another one is off the east coast of South America, and another one is off the southeast coast of Australia, between Australia and Tasmania. The one that's off the southeast coast of the United States is probably the most popular of the 3, because that species of kelp is primarily in a triangular area between Florida and Bermuda, and some of you kind of have ever heard stories and legends about the Bermuda Triangle, and a lot of things about the Bermuda Triangle. Now I'm not going to say that everything that you've heard about that is true, but some of the older legends about the Bermuda Triangle is very, very true, because during the time of transportation, when boats were primarily just sails, no motorization, the gulfweed, the sargassum kelp, is free floating. It does not have to have a root system and be attached to the bottom. So you had a huge area off the southeast coast of the United States, thousands of square miles, that was this floating kelp that may have been 1 foot to 2 foot thick, floating on top of the water. Well, you can imagine if you had a sailboat and you get stuck in the middle of this thing, if you ain't got enough wind to get you pushed through, you're done. And that's why the legend kind of got started on the Bermuda Triangle, was primarily, and if you look at some older maps, and I think I got a picture of one up here, if you look at some older globes or older maps, that area it'll have listed as the Sargassum Sea, and it's named after this particular species of kelp, okay? There's a picture of some bladderwrack. There's another one of the wrack families. There's an underwater picture of like a forest of giant kelp off the coast of Florida. I mean, the giant kelps out there, they say they can get up to 100 foot long, anywhere from 30 to 100 foot, and then they have to be attached to the bottom, but they're thick, as you can see there. There is a forest of giant kelp off the Channel Islands, between the main coast of California and the Channel Islands out there. Now, when you see some of these plants, let me back up one slide. When you see these plants like this, it's like, okay, now how do these things stay erect in a water situation? Okay, because if you cut one of them off and you bring it, they're just limp. Okay, if we get a corn stalk and we plant a seed of corn and it takes off growing, it's growing toward the sun, right? And it's rigid, where it'll stand up on its own, okay? So, these plants, if you notice on this picture, these little round bulbs, okay? Here's a close-up picture. A lot of these kelps has these little bulbs in here, and these bulbs are filled with carbon dioxide. So it's like little buoys on the whole stem of that plant, and that's what makes it stand upright in the water that way, okay? Now, I want to go back to brown kelp, because that's the one primarily we use the most in agriculture. We have 2 primary families. You have the Fucasia, and then you have the Sargassum, which the Sargassum we done talked about. This Fucasia is the one that probably most of us is the most familiar with, okay? Everybody heard of Ascophyllum kelp? If you've got a bag of feed kelp at home, when you go home and you look at it, it's probably going to say on there, I'm about to say 100%, it's going to say Ascophyllum, or it'll say Ascophyllum nodosum. You see the second line there I've got there, you have, this is a very specific family. Ascophyllum is more general, even within the Ascophilium family, there's, I think, like 50 different subfamilies under just the term Ascophilium. So if you're buying a kelp, and I'll partly say this because if you're buying a feed kelp and someone is selling you a kelp and you say, well, what type? Okay? So this isn't just about fertilizer, but it also has to do with what you are purchasing as far as a feed type kelp too. Right?
No.
Nope, you just shut. Okay. Now, the Sargassea, the Sargassium is under it, and then Sargassium, there's actually two types. You have your Sargassium natans and you have your Sargassium flutans. Now, Ascophilium, it's primarily a cold water kelp. It doesn't like water temperatures over 58 degrees and less. I mean, it will grow in waters that's what should be freezing. Salt water, obviously, if it's moving, has a lot lower freezing point than what regular water does. But Ascophilium kelp primarily like cold water. They primarily grow along the continental shelf, along any of your continents where you have the colder water. It prefers more of a sheltered water, to where it's not just in the open ocean that way. It prefers like bays or fords. That's one reason why Norway and Finland has a lot of exports a lot of kelps, because they have so many little fords and bays all the way up and along down the coast. Scotland and Iceland has a lot of those type of formations too. And that's why you see so many Ascophilium kelps coming from there. Ascophilium kelps can get up to 30 feet long. A big plant. They do have to attach to like rocks that way. And typically they're a dark brown to an olive green color. Now, here's the kind of the thing where we're going to kind of cross between a feed type substance and where I'm talking about fertilizer type substance, what I call fertilizer. Ascophilium kelps is really high in the plant hormone auxins. Here after lunch we're going to talk about hormones, really plant hormones more in depth. You have auxins, Gibberellins, and then they have a limited amount of what we call cytokinins. These are your three primary forms of plant hormone. But Ascophilium is extremely high, especially Ascophilium nodosum. That specific one is the highest in the plant hormone auxin. Okay? So, when we're feeding a kelp to an animal, traditionally it's just been thought, well, kelp's kelp, it's a source of micronutrients and trace minerals. There's a lot of other properties in kelps that still being studied, even the effects on human anatomy and also with animals. Even outside the realm of the plant hormones, there's a lot to do with nutrition that way with your kelps too. Ascophilium nodosum, like I said, is primarily from Norway, especially the one that we use for the kelp grow. The kelp grow product is exclusively made from Ascophilium nodosum. Now, something else. When you're cutting alfalfa, okay, if you want the highest protein, when you get the window for the highest protein and your best tonnage is going to be at what stage of an alfalfa plant? Anybody? Brian?
I wasn't paying attention.
Huh?
What was the question again?
Well, if you're going to cut alfalfa, when's going to be your highest proteins and your best nutritional value of that alfalfa? What stage of the alfalfa plant is that going to be?
Pre-flower, right?
Pardon?
Just point of pre-flower.
Early.
Early, yeah.
Pardon?
Very vegetative.
Yeah.
And just prior to what?
Reproductive?
Right, which is what? Like, I've heard a lot of people talk about like a quarter bloom. You've seen first blooms,
Yeah.
early bloom stage, okay? Now, if we're wanting the highest level of a plant hormone, in that particular species of kelp. Does that mean that we can go out and just harvest kelp anytime we want to? See my point? If we got a specific stage that we know for alfalfa, okay, this is going to be the best proteins, this is going to be the best quality. I got a particular window, and sometimes it's just what, a matter of days that I got to get in there. I got to get this cut, okay, for optimum. That's one thing that we look for this Ascophyllum nodosum that goes in our kelp grow. We want to know the harvest time of that kelp, because there's specific harvest times or stages in the kelp when these plant hormones is the highest. Some companies will just go out and just harvest kelp, okay? If you're wanting it for just strictly nutrients, your trace minerals and things like that for my use or for my animals' use, probably a particular stage is not going to be as important that way. But when we're looking at the hormones, it is. I got a particular stage that I want the kelp harvested, and that's what I want to use for my kelp grow product, to try to get the highest level of auxins in that product from when we're using it on a plant. Does that make sense? There's my map. So, when you look at this, here's your Sargasso Sea down here. Now, you get your colder water temperatures. I mean, a lot of your Ascophyllum kelps is going to be coming from up here. Here's Iceland. Here you get your Norwegian countries. You get your northern Scotland. There's some stuff that does come from Ireland. There's a little bit of stuff. Probably here in the United States, probably most of what you see is coming from up here in Newfoundland. Acadia kelp pretty much gets all of theirs from up here. Thorvin pretty much all comes from up here at Iceland, okay? Just to kind of give you an idea when you're looking at this. Now, here's where your Sargassum is, all down in this area, and then this be just kind of the top edge of where there's some Sargassum down here off the east coast of South America that way, okay? Just kind of give you an idea on kelps that way. Now, Sargassum kelps. Right now, there's 150 different species known of Sargassum kelps, even though they are in very specific areas in the world at this point. They like warm temperature water. They don't want cold, they want it warm. That's why typically those areas and regions I was talking about is going to be more of your warmer waters in the world that way. They can be free floating. There is a few species that does attach to the bottom, and most of them are like a medium brown to a tan color, a real light brown color. Now, like I said earlier, all of the feed kelps that I've ever seen on the market is all Ascophyllum. I've never seen anything any different. Part of the reason is because there's not been a lot of, I guess you would say, push into the Sargassum type kelps for whatever reason. I never have really understood why. Here in the United States, part of the reason was our wonderful president back in the 1990s, down in our area, where we're right adjacent to Arkansas, there's a lot of other names that we use for that particular president in the late 90s. But he made an executive order that no kelps could be harvested in the waters of the United States anymore. Which primarily the only thing it really affected, because we didn't have any Ascophyllums, because we didn't, other than up into Alaska, there would be some Ascophyllums, not very many. But it mainly cut off anything, what little work was being done with Sargassum kelps, it ended it there in 1999 on any harvesting at that point. So that made it really tough on us. because we specifically was wanting this species of kelp for making our foliar supplement product. Especially this Sargassum, that way. So, it usually grows 6 to 12 foot long. Like I said, some of it is free floating, and it is primarily, your Sargassum is extremely high in the plant hormone cytokinins, some Gibberellins, and very limited in Auxins. Almost just a flip opposite when it comes to plant hormones as compared to your Ascophilium kelps, okay? I kind of put this up here to show you the two different polar ends of the effects that kelps can have when we use it as an extract or possibly even as a feeding substance on animals that way. So, there's a picture of Sargassum. It looks a lot different than a lot of your other species of kelps that way. There's another closer picture. You notice how it has even the bulbs, the floating buoys, you could say on them, really looks different on a Sargassum as compared to your other kelps that way. So, you may have any comments or questions on kelps, okay?
What was the political reason for outlawing it off the United States?
Environmentalists, wildlife type people, they were lobbyists. They were lobbying that there was a lot of sea life that was in the Sargassum kelp, nursery for turtles and fish and all this different stuff. There was never any formal scientific, almost kind of sounds like climate change now, on data gathered. They just like, it's there, we got to stop this. And this is destroying wildlife. And so they just ended it. But since then, there's some mood changing that's just started last year. But it was pretty much completely eliminated. No questions. I mean, it stopped.
Small chunk of the ocean's awful damn big.
And, well, where no studies was done, now, like the state of Florida is really beginning to question. The state of Florida is now coming back and saying, okay, you're saying all these turtles and fish is supposed to be out there. Has anyone decided to do a count? And there's not any scientific data. Well, so now there's some of them that's pushing some in the science research world. Go gather some information. We want to see what's really going on.
Keith,
So,
wasn't there a problem with, even down in Florida, that they're having all this?
oh yeah,
seaweed
you
come
kept
on
up with any of
beaches?
the news the last couple of years? The kelps coming on the East Coast shore and causing all the problems. That's one reason why Florida is kind of calling questions on it, because it's beginning to hurt their tourist money. Because this kelp is washing up on shore and it's got all these dead fish in it that get stuck in the kelp, and it's all washing up on shore, so you have these nice, it's hard to get someone to go swimming in the beach when there's dead fish and dead kelp strung all on the beach. And so now that it's affecting Florida's tourism budget, now Florida's like, okay guys, what's going on? Is there really anything to this? And everybody's like, nobody knows. So the Lord, it's interesting, the Lord's really opened up some doors on some things that's occurred. And it's really interesting what He's doing. That's why I'm a little bit distracted since last September with a lot of the fertilizer things, because the Lord's opened some doors, opportunities for us to be involved in this Sargassum thing on getting all restarted and reinitiated this way, which would greatly make a difference, which I don't know, Brian and I was talking about this, about 2001, is that when you kind of got started, Brian, with the zone thing? See, the last years that we really had a high concentration of sargassum in our foliar supplement product ran out in about 2001, 2002. So me, I remember it. I mean, used to, the foliar supplement was almost kind of an orangish brown color, where now it's a dark brown. We tried to make the best do with what we had, which wasn't the ideal, but I'm excited right now that, hey, maybe in a year, we can be back to where we were 25 years ago. That I'm really excited about that way. So, but yeah, it's where sometimes politics, this is kind of the thing now. Brian and I was talking about the electric vehicle thing last night, and some of the brain had some things on some of these ideas that some people come up with, and it's like, okay, do you have any facts for this? Well, no, not really. It's just a hypothesis. I mean, it's not even a theory at this point. But yes.
Why is kelp so hard to get right
I don't really know.
now? Like the feed kelp.
I don't know. It don't really make sense. I think part of it is, as you seen from the map a while ago, I'm trying to get there very quick. Europe is really beginning to catch on to some of the things here. I was talking about the Asian people eating more kelps. In Europe, the last 10 years, kelps is becoming a little more of a, had you used the term, delicacy in Europe. And I think that some of the kelp that's been harvested up here, that primarily the United States, has been the main outlet for all of that. That I think some of it's getting diverted and staying in Europe and going to some other countries at this point. I think that's what's going on. I've asked different people that I work with and that I've known in the kelp industry for a long time. No one can really, I asked the same question at this time. I don't really know.
Somebody told me they had a bad harvest.
No. There for a little while during the COVID thing, transportation was a big deal. Because where all of it was having to come across the ocean, where a lot of the ports and a lot of some of those issues was going on, which I know there was in some places. And there still is a labor, I mean, even just out, you hear people in the workforce, I mean, labor is a big issue this day and time. I don't know if that is kind of, I don't know. Sometimes I wonder if the supply is being kind of cut back a little bit in order to run the price up, because as you know, the prices went way up over the last 4 years on kelps that way.
I seen a documentary on this families that were living like Indonesia or Philippines, and the generations, they all lived on boats. They never came ashore. And now they're making their living collecting seaweed. But this was, you know, something they.
There is
So
some.
that's probably what they're into, the kelp, in other words.
There is some countries, especially in Asia, that, I forgot the term that they're using, actually someone that's kelp farming,
Yeah, that's what they.
that's getting to be a little bit of an industry in some countries that way, where people are specifically cultivating kelps. They get a harbor area and they're specifically cultivating and doing things to try to protect it, where they can come in there and have a bigger harvest, which is kind of like what we do with plants that way too. Yeah. Anybody else on kelps? Anything else? Good questions. That way. No one does, then I'm going to let me switch here real quick. There we go. I get the progressive Now, I'm not in dairy country, so I get the progressive cattlemen. Probably some of you have seen the progressive dairymen, progressive cattlemen. We get some really interesting cartoons sometimes that shows up in that. Okay, hormones. Okay, now, hormones are communicators. Each one of us has a vast number of different type of hormones that functions in our body. Just to pull one out, insulin. Insulin is a natural hormone. Probably one that's common enough. Most of us knows what the purpose of insulin is in our body for regulating and monitoring blood sugars. But our body uses hormones to communicate with other parts of the body. Our body doesn't have an intercom system where our brain can get in contact with the liver and different things. That the body is primarily using hormones to function with other parts and to be able to communicate with those parts. Okay? That applies to us, it applies to cows, it also applies to plants. Plants use hormones very specifically in even determining how many leaves is on a plant and where the leaf is going to be at on that plant. A hormone does that. We can throw all kinds of nutrients into the soil and into plants. I mean, I'm a firm believer of foliar spraying nutrients. You can foliar spray nutrients onto a plant, but if a plant does not have a proper balance of hormones, the plant does not know what to do with any of those nutrients and cannot function correctly. Okay? This is something that I growed up around pretty much my whole life of farming. Okay? Talking about plant hormones and how hormones function. There was very limited information until just really the last 5 years that there's actually, the scientific world is actually going back and doing a little bit of studying with hormones. And it isn't that the hormone thing's new. Okay? Auxins was first discovered back in the 1940s. Okay? Long time ago. Cytokinins, discovered back in the 1950s. And it's just like once the scientific world found them, then it's just like, okay, and we walked off. Didn't do anything else with it. Okay? But they are the communicators in the plant. Okay? There's 3 known hormones, and I've got on there at this time. I believe there may be more. I mean, when you talk about human anatomy, and as they kept exploring the human body and figuring out how the human body is working, which I don't think they completely know even that at this point, but there's 3 known hormones: auxins, cytokinins, and gibberellins. Okay? You get out in California, you'll hear people talking about gibberellins, gibberellic acid. They use it a lot on table grapes. Okay? Gibberellins is really good for making really nice big grapes. Okay? But if the humidity and the temperature don't cooperate and you go out and apply a plant with a synthetic gibberellin, it'll turn all the grapes sour, bitter. Okay? So it's a synthetic. Okay? It's the difference between a synthetic and a natural form. Alright? Keep that in mind. Now, got there, what do they do? Should we be concerned about the levels in plants? And also, can we do anything to change these levels? And we can, that way. Now, I want to talk about auxins here first. There are 5 natural occurring indigenous, I'm going to use I put that in there specifically. Means indigenous means that they are naturally in a plant. Any plant, you're going to have these 5 auxins in a plant. All right. Now they use abbreviations for these. This one's I3A, and then you have 4,3AA. So they use abbreviations for a lot of these. When you look at the length of some of these words, then it's kind of like, I'm glad they use abbreviations this way. Okay. Now, when we talk about auxins, some of you maybe glance on this in the past in a meeting, there's two forms of auxins that probably most everybody here has heard of, okay? There's two synthetic ones. The first one's 2,4-D. That's a condensed abbreviation like the ones above. I put it on there that way because most farmers recognize that. 2,4-D, it's a herbicide, right? That is a plant hormone. It's a synthetic auxin. Now, I've had people argue with me, well, this thing and these plant hormones, you can't do anything with a hormone that you're going to have an effect on a plant. So, you're telling me that 2,4-D don't work. Does anyone, 2,4-D is one of them things that you gotta watch when you use it, because if it drifts over on the neighbor's field, you can have a big problem, okay? There's another one there, 2,4,5-T. Most of you are probably familiar by the more layperson name, military code name, Agent Orange. Agent Orange is 2,4,5-T. A lot of us are familiar with those too. Now, do auxins work? Okay. You say, okay, we're supposed to be using these auxins, and that's supposed to be helping me with my farm. You're talking about two of them here that will fry everything. Okay? When you look at how auxins work, and the scientists were really smart in what they were doing when they come up with this 2,4-D and 2,4,5-T back in the late 50s and early 60s. Okay? What they were doing is, whenever you take a 2,4-D and whenever you put it on this plant, it makes the plant grow itself to death. The plant, whenever it takes that 2,4-D and it starts that, the metabolism of that plant speeds up so fast that it can't keep up with the pace, and the plant just literally internally explodes and dies from the inside out. Okay? That's how 2,4-D works. That plant hormone, that's what it does. Okay? When we look at auxins, auxins is really important for the cell wall integrity. The cells in that plant, the structure that gives that plant, we talk about corn growing and having structure. All these cells that's filled with fluid, and there's fluid on the outside, so how do we get this corn plant that's rigid? Okay? It's each individual cell and the structure of that cell and how that cell wall is made. Okay? Auxin is the big determiner about that cell wall. Alright? So remember that. How big that cell is going to be, either length or width or whatever, it's three-dimensional. Whatever that size is going to be is determined by auxins. Okay? That's why auxin is so important. That's where, like 2,4-D, it weakens that cell membrane, and more or less, you ever blow up a balloon, and you keep blowing, and you keep blowing, and you keep blowing until finally it's going to pop, right? There's going to be some spot on that balloon that cell membrane gets too weak, too thin, and boom. That's what happens when you use something like 2,4-D or 2,4,5-T. It weakens that cell membrane and the pressure on the inside of it, and it just blows up. Okay? But that is supposed to do that in an elastic type situation. And that's the importance of your auxins that way. Like I done mentioned earlier, that's primarily the thing that we're focusing on with the Kelp Grow product. We're specifically wanting high auxin levels and high active auxin levels to where when you take that product and you apply it, let's say, to alfalfa, that we're going to be able to get bigger stems, thicker leaves, okay? Some plants you'll end up with bigger diameter leaves, not necessarily alfalfa. I've seen that a little bit in alfalfa. I've seen more of the thickness of the leaves, more so in alfalfa with auxins than necessarily the size. But the plant hormone auxin in the kelp grow is the specific thing that's doing those type of things. That's one reason why I was talking about, when I was talking about the harvest time of the kelps. So I want, when that kelp is harvested, I want the highest auxin level in that kelp. When they harvest it, for whenever I do the extract for the kelp grow, that I can get the highest auxin levels there. As far as I know, no state has a registration process or registration step for putting a guaranteed auxin level on a label for a fertilizer input. That's why I've never put it on the label. When we first started getting into registration with states, we kind of threw that information out to the state registration people, and they're just totally clueless. What's this? That ain't in our book. You can't claim that on a label because we don't know what it is. Okay, so we pulled it off. We specifically at the beginning was not wanting to do our NPK, but the traditionalist fertility inputs is all based on what? NPK. So that's the primary reason when you look at this product, I mean, we're only 0.3% nitrogen, 0 phosphorus, and 2.5% potassium, and you're wanting me to put 12 fluid ounces per acre on? Well, that's not a lot of NPK. How can this product do anything? Well, if you're just looking at it from NPK, nothing. This product isn't going to do anything. But it's all based on the hormone aspect that way. Okay? You want to have any comments or questions about kelp growth?
Yeah. When stage of growth to apply it and method of application.
Okay. Method of application primarily, probably in this setting with this group, is going to be either like on corn or other things like in a row starter or liquid row starter, you can put it in there, or foliar sprays, like I was talking about like alfalfa or whatever, like.
Would it be more in the vegetative, early
When,
vegetative?
and the thing is, you're wanting to do this specifically when you're wanting vegetative growth, okay? I mean, it will make a plant grow. I've got some produce guys I'm thinking about that, especially greenhouse tomatoes, when I've got people that are trying to start greenhouse tomatoes here next week, okay? A tomato is day-length sensitive, okay? The days are still really short here. I mean, even if you go down into my part of the country in Tennessee, it's still too short for a tomato. And so these plants don't want to grow, they don't want to do anything. Well, I've got people that's figured out, hey man, I can put this kelp grow in here once or twice a week, and I can get these things growing. Well then, as we was talking about in our zone dealer meeting, one of the dealers was talking about different things, and he said, you know something, he said, I found that most farmers are morons. The more we put on, the better we think it is, okay? So I got it, so, yeah. I mean, so I say that in that relationship is that I get someone that sees something like kelp grow work, and they're like, oh man, I can put this on them, and these tomato plants, these pepper plants, and stuff takes off growing, and then they want to keep using them. And then they call me later, and it's like, I can't get any tomatoes to set. And I'm like, how much kelp grow are you using? Well, I'm spraying it 3 times a week, and they give me rates, and I'm like, you're never going to get a tomato on that plant. Well, what do you mean? This is Auxins. This is telling a plant to be more vegetative inside. Remember I was talking about a while ago? Bigger stems, thicker leaves, okay? Now, if you've got a market that you've got someone that's wanting to make salads out of tomato leaves, you can use this all the way through. But most everybody wants these round red things, and you're not going to get it with using a lot of kelp growth, okay? Now, flip side of that, if you've got alfalfa, all right, you want to, as we was talking about a while ago, for optimum nutrition, we want to try to keep alfalfa before it goes into a complete reproduction stage. So, using something like this, a lot of its life, if you want to go in and do multiple foliar sprays on one particular crop, it's going to keep it from wanting to kick into bloom stage. So see what I'm saying? We can naturally manipulate these plants that way. Okay? That makes sense? So, if you get a plant that is supposed to be going into reproduction, I got the same thing happen with people with green beans. get to applying Kelp Grow on green beans, and they get these great big old biggie bushy green beans. I can't get, my wife's complaining, I ain't got any green beans, I don't know if this program works. It's like, cut the Kelp Grow out, no more, okay? So, Ryan?
I think Bob was maybe asking, if you look at like alfalfa compared to soybeans, compared to corn, when would be the time to foliar feed it as far as the growth?
First by foliar beans and then.
Soybeans is going to be really early, especially up here where your earlier group beans are kind of blooming over a lot longer span and starting earlier. We really want to watch what you do with this Kelp Grow. Now, I've had some people in this part of the country where they think their biggest limiting factor on their bean yield is because the plant's not tall enough. We may want to put a little bit of this in like a row starter, not necessarily doing a lot of foliars later on, to try to get some quick growth to stretch that bean plant up a little taller, okay? Now bean plant, where they're doing more blooming, we want to really be careful with this, okay? Now, corn, even though corn does go into a reproduction stage, corn doesn't seem to be as sensitive to this. Now, if you get the row crop guide over here, like the 28 to 32 day foliar spray, so in this video we talk about that stage when the tassel is forming, I don't want to use none of this, okay? I want to keep it all reproductive. That's where we want to only use the foliar supplement, no Kelp Grow. Now, let's say either earlier than that or after that, if you've got silage corn, where I'm wanting thicker leaves, more structure, you may want to come back in with a later foliar spray and hit it with kind of an equal amount of foliar supplement and Kelp Grow. Okay? Does that make sense, Brian?
Most of the time, aren't we using the Kelp Grow along with a little bit of foliar and not Kelp Grow alone in most of those situations?
I always, when we get later in this hormone thing, you'll see it. If you look at the program guide over there, I never have Kelp Grow by itself. We've never done that, okay? As you'll see, we always want at least a 50% rate of foliar supplement to your Kelp Grow. I said at least, okay? Let's say we're doing alfalfa, and we're wanting this alfalfa to be very vegetative, okay? I want to use, let's say you want to be really big, I want to use 24 ounces of Kelp Grow per acre in a foliar spray. I'd like to see 12 ounces of foliar supplement with it, okay? At least 12. The reason being is we'll look at it in a little bit, we need that backing of what's coming through that foliar supplement, okay? I never want to use this by itself. Now, we'll use the foliar supplement by itself, but never Kelp Grow by itself, okay? That makes sense?
What happens if you do?
Well, as we'll see, the cytokinins hormone is like a, best example, me being a criminal justice background, is like a traffic cop. If you get at a busy intersection, had an accident, usually you got a police officer out there, and I've seen some of them, they're lousy directing traffic, that they're directing traffic to keep things moving, okay? Cytokinins does that in the plant. It's directing traffic. It's telling Auxins where to go. It's telling Gibberellins what to do. It's pointing nutrients to go different directions, okay? So that cytokinin is a base core of the main communication system of a plant. So that's why I always want a little bit of foliar supplement for your cytokinin base to give the directions, okay? Does that make sense?
Well, it would be good in the nursery business.
What now?
If you were selling plants for people to take home with their garden, it'd be great for that because you give good looking plants.
Last week I did a presentation in Indiana, and there was a gentleman there that he and I was talking about it. I guess it's been about 6 years ago. I was told to sit down and lay out a program for corn and alfalfa for him. So I laid out this program. And he'd been with another liquid fertilizer company, and he'd been using a lot of dry. And when I got done, he told me, he said, "This won't work." I'm like, why do you think it won't work? He said, you're not using enough NPK. He said, you can't raise a 250 bushel corn crop, because it's down there, more grain area. You can't raise that with the limited amount of NPK you're using here. And I'm like, well, it was in a situation where he was getting pressure from some other owners and partners. So it was one of them things, he just kind of grinned and bared it, and he put a small plot in of what I recommended. And when we got done, before he started planting corn, he said, are you sure you don't want to add anything else? I'm like, nope, do it just like that. I tied for the top yield, but I had 50% of the expense as the other one that we tied with. Then he was like, okay, we need to do this again. I don't think you can do it a second time. And we did it in the same plot area. He done it again. Tied, and I was 50% of the cost. And he's like, I don't understand it. Well, I did this presentation down there where he was at, and I made a comment. I kind of brought it up. There's guys there, and he just started laughing. And I'm like, he's like, you're 100% right? Because when I left that day, the first meeting, one of my sons was with me, and I said, we'll never hear from this guy again. And he started laughing about it. And he's like, he said, I still don't understand how you do it, but he said it works, and that's all I got to know. And he just kind of goes, it's kind of a faith thing that he goes on from there. But I say that in the sense that if a plant gets all this NPK nutrients that we throw at it, and there's not a proper number of traffic cops there to direct things in the plant, then you get what? A traffic jam. Nothing moves, right? If everybody's clogged up at this intersection, we can't get nothing moved, so what's the use of having all of it? That's what helps when you have the hormones, that you're keeping things moving efficiently and throughout the plant. Then a lot of times we're applying NPK fertilizer, agronomy, and you get someone that really will be honest with you, a traditional agronomist will admit this. We realize there's an inefficiency factor with nutrients in plants, so we just over-apply to try to make up for the inefficiency factor. Well, why don't we just make it more efficient, then we won't have to buy as much nutrients. Okay? And that's what we're trying to do with the hormones that way. Does that make sense? I've seen another hand. Yes?
So this could be put on with the same tank mix as like N-Fix and electricity, you put that all together?
Yes. I would prefer it to be that way. Because all those things have a specific reason and purpose, and where they function together, it will help the whole thing work better.
And also have foliar spray, the foliar besides this, all the same tank, right? You can mix that all, like kelp,
Yeah.
foliar, yeah.
Yep, it can be all in the same thing, yes, that way.
Sure.
The only time, I mean, it's like we were talking about the growth thing, that we don't need any kelp in there, but as far as the recipes calling for everything, yes, we can put it all together in one tank mix. Okay. The only thing, if you're conventional, the only thing that's not supposed to be in the same tank mix is like 3-18 and liquid kelp. Okay? That way. So, I see another hand. Yes.
One thing to note though, when you're mixing that, you watch how you mix your kelp or your sprinkler plug if you don't have diluted enough. I've had that.
That's a pH thing.
That's a pH
Okay.
thing, but it can happen.
Yes, that is true. I'm glad you brought that up. The Kelp Grow product is going to have a pH of about 8. Okay? And so if you get something, especially conventional guys, and let's say they're going to be out here spraying alfalfa and you're doing Kelp Grow and you're using liquid cal, the conventional liquid calcium. That liquid cal has a pH of 5. And this has a pH of 8. You put them two together, and if they're not diluted enough, you're going to have black cottage cheese. Okay? Now, you did not damage either one of them, that if you get enough water and agitation, they will go together. You didn't hurt anything, but just the two polarization of the two pHs, and you stick them in the same room together, we're going to have problems. You'll get some curdling that's going to happen. Okay?
So
I'm glad you brought that up.
You gotta use more water.
You put a little more water in there, or if you will dilute one of the two of them, let's say you have the Kelp Grow and you dilute it 50% with water before you put it in there, you should be okay. Another thing is, kelp grow and like TR3 or VR5. You don't want to dump TR3 or VR5 in a bucket straight with straight kelp growth. You're going to have black potted cheese. I mean, it's going to happen that way. Okay, it's going to curdle. But neither one of them is damaged that way. Okay? It's an excellent point. Anybody else? Okay. Ryan? Yep, I can stop. This is a good stopping point. Okay. We were talking a while ago, what Brian was talking about really fit in good with kind of what we did on this hormone thing. When we talk about different plant hormones, we talk about auxins and how auxins function, 1 to 3. On any plant growth regulators that's regulating plant growth kind of falls in this category, but especially in the area of, like I done mentioned earlier, about stimulating cell elongation in stems and in leaves that way, and also plays a key role in cell division and what they call differentiation, as far as on the separation between cells and how cells function within a plant. And also in plant development, not as much as far as plant setting, but more in the developing of a fruit that way. We talked about earlier as far as like on soybeans or tomatoes, on auxins making a plant be in more vegetative growth, not necessarily setting that bloom and setting that fruit after that bloom is pollinated. Now, like the example of a tomato, once that tomato, that fruit is set, and you get your little tomato starting to form, then auxins plays into a big factor in how big that tomato is going to be, okay, on sizing the diameter of that. So that's why you get some crops, tomato prime example, that plant is constantly, through its growth season, is constantly going from dominant auxins to dominant cytokinins. It's going from more plant growth to fruit set. So it's kicking in and out of reproduction cycle continually throughout the season, and maybe doing multiple things at the same time, okay? Soybeans would be kind of fall in that same type of category. Now, early on, a soybean plant, kind of like a tomato, is putting a lot of emphasis on plant size and plant structure, to where auxins plays a big factor in that. Also, auxins is real important for root formation. Now, there's a statement here that auxins is going to inhibit lateral branching, which they call apical dominance. That is talking about how quick, probably the best example to this is a pepper plant. You had any bell peppers or any type of a pepper plant? As far as that plant growing, and you get a certain amount of growth of that stem, and then you have a growing node to where 2 leaves come out, okay? Auxins is going to inhibit lateral branching, a branch or a leaf coming out the side. So if you get too much auxins, you're going to have a really long stem or growing node in between each set of leaves or branches, okay? That's what that's saying. It's going to inhibit lateral branching coming out from the main stem. Now, gibberellins, the second one of your 3 plant hormones. I'm not going to spend a lot of time on this. It's known, they're all acidic forms, gibberellic acids. They're involved in the natural process. And one of the key things that really interests most of us here is involved in the breaking of the dormancy of a seed. Okay? Have you ever thought about when you take a corn seed or a soybean seed, it can sit here in dormancy in a sack, especially if you put it like in cold storage for years, decades, centuries. And you can take it out and plant it, and then it knows it's supposed to germinate and come up. Okay? Now, you get temperatures, you get moistures and light, all those do play a factor in signaling that seed, okay, it's time to sprout, it's time to come up. When those factors get in place, Gibberellins is the first thing that gets activated, especially out of the plant hormones. It's going to start this process of breaking the dormancy in the seed and starting the whole germination process. The whole thing of breaking down where a seed contains starches, long-term energy, or hydrogen like Brian just talked about earlier, of breaking that down to break down the starches into more simple sugars for this plant food, because until that plant gets up out of the soil and starts the photosynthesis process, it has to have a supplemental energy source. Gibberellins is the one that plays the factor in that. Matter of fact, in our type of farming that's going on here, this is probably going to be the most important phase of where Gibberellins plays a big factor, is how seed germinates and the energy of that plant when it comes out of the ground. So that is primarily going to be determined in setting that seed when the seed was produced. The previous generation, okay, is going to play the big factor in how the amount of Gibberellins is in there, okay? Now, cytokinins. Now, as I go through talking about cytokinins, I've got here kind of parentheses here CK. So anytime you see this CK showing up, that's referring back to the term cytokinins, okay, or other plant hormone. Cytokinins is important for cell division and also apical dominance, as we'll look at here in a little bit. Also for shoot initiation and growth, and also the arrangement of the leaves. How leaves end up on a plant, which side of the stem, all of that is determined by what's told cytokinins to do, okay? Now, the genetics, the DNA that's built within that plant is the blueprint, but then that blueprint is being read and implemented by cytokinins, okay? Also, it's very important for the vascular bundles. When you get the stem of a leaf, the little holes and pores within that stem, as far as where water moves and nutrients moves up and down, cytokinins is important for that. And also, the aging process of the plant. Cytokinins determines when a plant leaf is terminated and it falls off. We just came through fall of the year. You get to the fall of the year, the temperatures start cooling down, the deciduous trees start turning colors, and the leaves fall off. Cytokinins is the one that determined all of that. Okay? That's how important this hormone is. Whether if you get an early die-off of a plant and how it's going to age. Seed germination, and here's a very critical point. If a plant is short in cytokinins, it's going to have more troubles with stress. If we have adequate cytokinin levels, the plant is more flexible. It can maintain and be able to fluctuate with internal and external stresses, whether we're talking about drought, hot, cold temperatures, too much water, all these different types of stresses that's created on a plant. Cytokinins is needed to help this plant be flexible and be able to do these things, okay? So, earlier when we was talking about, Brian brought up the thing when we was using Kelp Grow, I said I always wanted what? A 50% rate of the foliar supplement to be with the Kelp Grow, because I said I was wanting cytokinins there. This, you kind of see this thing with the internal and external stresses. Any stresses that this plant is coming up on, also on how the plant metabolism is functioning, is dependent upon cytokinins. Okay? So, now, I put this in here because originally I thought we're in the process of actually changing the name of the foliar supplement product. And this is gonna be the new name. Everything about the product and everything's gonna be the same as what we've been doing. I see it was a good time to jump in and to make a name change. And the reason being is, through the years, the name, the foliar supplement has been kind of a hurdle sometimes with people. That whenever someone like Brian or myself makes a recommendation to you, say, okay, you need to use so many ounces of the foliar supplement in your cornstarch. Well, then people kind of get puzzled, and it's like, well, I thought you said it's for foliar supplements, for foliar spray. Why you want to put it in the starter? You see what I'm saying? I've had people come back and call, having issues later in the season, especially like with produce or something, and I'm asking, okay, how many, what's your rates, what are you applying? And then I'm like, did you not put any of the foliar supplement through the drip line? Well, no, I thought it was only for foliar sprays. It says it's a foliar supplement. See my point? And I've wanted to change the name for years because it's kind of created some issues. I don't know if any of you is probably going to see this label this year, probably next year, probably so. But we're kind of in this process of making this change this way. Now, what I was talking about a while ago about cytokinins playing an important part on apical dominance. When you look at this, this is a picture of a soybean plant. When we get this here, this is, we've got a stem here and for leaves, but here's the main growing point right here, okay? Especially right in here. This is going to be another leaf or stem, and here's another one over here that's starting to unfold and go out, okay? This center part right in here is where a lot of decision making is being made. Where there's going to be a leaf out, how much more growth we're going to have before we're going to send out another leaf, okay? All those decisions is being made there on the go, okay? This is kind of a crosscut section of what they call the SAM, or your shoot apical meristem. That would be that very end that I just showed you, where it's growing. Well, when you look at this, here's CK, this is cytokinins. Here's cytokinins. Here's auxins, okay? You notice there's no gibberellins here at this point. That's not a factor. But if you notice, the little red bar, kind of a T-looking thing, it's saying suppression. The black arrow is activation. Do you notice that, usually there's a saying, all roads lead to Rome, okay? That when you look at this, whether if it's an activation or if it's a suppression of an activity, no matter who it's with and what, they all end up where? At CK, right? It all has to do with cytokinins wherever, other than this very center part. This is more like, this is your DNA part as far as on making those decisions. But if you notice, Auxins is over here. He can't do anything until cytokinins has him do it, okay? Now, this is a potential leaf. This is where an actual compound leaf is trying to come out, okay? Cytokinins suppressed or stopped any movement to this side, but it let movement go this way to do this leaf. So this just shows you this diagram of where cytokinins, I was talking about, is the traffic cop, is determine who gets to move, who has to stop, and what happens. That is what cytokinins is doing here, okay? That's the reason why, if we're doing a foliar spray or whatever, if we're dealing with the plant hormones, we always need a certain amount of cytokinins in that mix, just to make sure the plant has what it needs to be able to regulate all the other nutrients and all the other hormones, okay? Here's another kind of picture of that apical dominance area where you get a new leaf shoot coming out and a lot of that. And they kind of have this cut where you go farther down the stem. And then here's a cross section we don't really think about a stem that way. When you look at it at the end with our naked eye, we can't really see a lot of the pores and a lot of the spaces that's in there. If you get a magnifying glass or especially a microscope, you can kind of see some of these vessels and air spaces that's here. Cytokinins is what's determining that structure. How big the vessels are, how big the bundles are, as far as how much nutrients and water can be moved up and down this stem. Cytokinins is critical for that. That's one reason why, let's say like a corn starter, that's why I want the foliar supplement high in the cytokines right there at that seed. Remember earlier I had on there that cytokines is important for on germination. I want it right there at that point. And then also it's right there close by for the roots to start absorbing early to help in all this early development of a stem for a bean plant or even the shoot for a corn plant. Okay? Your growing point on that corn plant is very important there. Okay? Now, leaf size. We start talking about the diameter, the length. We talk about like a corn or grass family, how wide that leaf blade is, how long it is, how thick it is, okay? A lot of these things on this size is regulated by cytokine. Now, I'm not saying that it creates some of it, but it's regulated, all right? Cytokines also shorten the transition between different phases. So like you go back to like that tomato plant or a soybean plant where we're constantly going from bloom to development, going back to bloom and development of the plant, okay? How long it takes that plant to cycle is determined by the amount of cytokine levels that's there, okay? Also, and then this goes back to the thing of what I was talking about earlier with the 2,4-D. Okay? Cytokinins increases the cell pressure while auxins promotes cell wall loosening. I was talking about earlier about how 2,4-D will loosen the cell walls to such an extent they get so thin that the cells will just rupture. Okay? The natural process that's supposed to happen is auxins is supposed to come in, loosen or weaken that cell, and then cytokinins comes and increases the pressure on the inside of that cell, which more or less blows that cell up a little bit bigger. And it's supposed to do it in a repetitive stage between cytokinins, auxins, cytokinins, auxins. 2,4-D jumps past the cytokinin stage and it just loosens the cells and they just fall apart. Okay? There's no cytokinins there to regulate that. Now, when we look at the cytokinin function, now this is more like a broadleaf, like a soybean leaf here, okay? And it's interesting, a broadleaf, your dicots and your monocots, which your broadleafs and more of your, we'll say your cereal grains, the way the leaves grow is totally different, okay? When you take a broadleaf, they're kind of going through all three stages, or not necessarily that stage there, but they're going through these two stages almost simultaneously. They tend to grow around the outside edges and from the inside out, to where a cereal grain, corn or a grass, tends to grow from the bottom, and it's more or less literally like we kind of think. It's just growing out, okay? Not as much both directions like what you get here. Now, if you notice, I'm not going to go through what all these different interactions are, but if you notice the proliferation stage, the expansion stage, and then this leaf senescence, that's the biological term for the process of starting to die. When the plant is going into the senescence stage, it is in the process of dying at that point. Now, you notice cytokinins is playing a major role even all the way to the end of how the death of this leaf is going to occur, when it's going to occur, and what's going to occur. And you think, okay, if the leaf dies, the leaf dies. Well, that's not necessarily true. Here's an example. This is an experiment, and it's interesting, some of the latest information that's being really kind of worked on with plant hormones is dealing actually with agroforestry. It isn't about rubber agriculture, it's about growing more trees and growing trees quicker. That they're actually doing some studying now on these plant hormones. They found that a shaded plant, if you take a plant and it's in a shaded condition, you have a significant decrease in cytokines in all the plant leaves, and it's going to make a difference on the size of the leaf. We was talking about a while ago about greenhouses and raising produce this time of year, okay? If you're trying to raise a plant this time of year, sometimes it's just like, man, I can't get, this plant just isn't as big. I plant the exact same seed and the same variety in my garden in the summer, and I get this bigger plant, okay? It has to do with sunshine and the length of light that it's getting, okay? So in this experiment, they intentionally took some plants and they put them in a shaded condition. They noticed the cytokine levels dropped in the leaf, and the leaf size got smaller. Then also, because of not only the size, but also the number of leaves is going to be decreased. But when they took that same plant, still in the same shaded condition, and they started treating it they used a synthetic cytokinin here. They treated it with a synthetic cytokinin. They was able to get the leaf size to go right back to the size that it should be for a normal light condition. Which, it was interesting when I was going through and reading through all of this research data that's been done the last 2 or 3 years, it really wasn't anything that I hadn't seen before, that I'd actually physically seen with crops, where farmers like you doing things, putting the foliar supplement or doing the Auxins, the kelp grow on a plant and seeing results. It just helped me kind of explain how and why it's good. I mean, I have seen that before. You get someone in a greenhouse this time of year and doing extra foliar supplement, it seemed like, well, my plants is bigger. They're advancing quicker than even what, let's say, a neighbor that's not using the foliar supplement has the same variety, planted tomatoes, you started your plants in the same house, but yet you got to market a week earlier than what they did. Well, most people, what's their question? Well, what kind of fertilizer did you use? How much potassium did you use on the tomatoes? Okay? That tends to be the questions. We don't really ask as much about, well, what type of plant hormone did you use? Okay? Now, this is something important that they brought up that's really important, especially if you are like corn and soybeans. Whenever it's time, this leaf senescence, when you start having the die-off of a leaf, color starts changing in the plants at the end of the season. Cytokinins is responsible for the disassembly of chlorophyll, proteins, lipids, and pigments in the leaf. Okay? So when that leaf, let's say a soybean plant, you get to the end of the season and you start having color change. Starts going from green to a yellow, okay? Cytokinins is actually breaking down and tearing down house, disassembling things, this leaf's dying, it's disassembling proteins, carbohydrates, even chlorophyll, and then it's dispersing, it's intentionally dispersing all of this energy and nutrients from that leaf, and it's dispersing it for the development of seed and fruit. See what's going on? If that cytokine level isn't high enough, all that energy that was built from the photosynthesis process in those leaves stays in the leaves. But if we have enough cytokinins, that plant has the ability to pull some of the nutrients from that leaf and take it and put it in that soybean or put it into that ear of corn. Okay? It can translocate those nutrients. Those nutrients aren't just stuck in that leaf and it dies and it falls off. Okay, that's where it's at. Okay? Does that make sense? So cytokinins is very, very important for that. We kind of talked about this internal and external stresses. So whenever we get a reduction of the cytokinin levels, that's when the plant is determining that, okay, this is getting to the end of the season. So if your cytokinin levels gets too low, the plant may naturally think, well, if you're at the end of the season or not, say, well, it's done. Party's over. We're going to start shutting down. So that's where the cytokinin levels can be a big factor in how, if you have early premature die-off in a plant. Now, something else, when we start talking about forages, especially if you are specifically using more improved type forages, if you're intentionally selecting breeding of forages of trying to get more sugars and more proteins, okay? Cytokinins is also importing elevated levels, is also important for carbohydrate metabolism and also stimulating the energy process, or more or less speeding up the photosynthesis process. That if we have a plant that has the genetic potential, a grass, or let's say an alfalfa, that's been bred into this to be a high energy, high protein, high digestible protein type of a plant, if we elevate the cytokinin levels, then it's going to help that plant get its full genetic potential in getting you the most protein and sugars into that forage or into that crop. Does that make sense? That's the reason why I always want at least a 50% rate of foliar supplement anytime you're using, let's say, kelp grower or anything else like that, okay? So we're kind of talk about that on the die-off. It also acts as far as on slowing the process on die-off off. Also, even though I said there earlier, when it's time for the leaf to die, cytokinins will break down chlorophyll, break down proteins. But whenever the plant is actually functioning, cytokinins does the opposite. It prevents proteins from breaking down and chlorophyll destruction that way too. So it does both. It's determining whenever it's time for it to break down, it's going to do it. But whenever the plant is supposed to be functioning, it keeps the whole process going. Also, on stabilizing proteins and chlorophyll in that plant is really important with your cytokinin. And then the last thing, plant immune system. It's going to help give that plant more resistance to disease. Okay? Now, listen to what Brian was going through up here a while ago. I heard a little bit about the redox thing. I've got a friend that's kind of Springfield, Missouri. They're kind of their, how would you say, environmental monitor person over the water and the sewage and things, doing a lot of this checking. I've heard him talk about those numbers, but I'm kind of interested to see where, of checking some of the redox numbers on some of the, like the foliar supplement and the kelp grow and some of them. I've kind of listening to what Brian is saying, it's kind of got me wondering, knowing that cytokinins in the foliar supplement helps the plant resist disease, okay, where's that fit into all these numbers that we was talking about a while ago? It's going to be, I'm going to do some experimenting with that, that way. Now, when I've got MS there, I'm talking about MS crystals, okay? I was mentioned earlier about the thing about farmers, sometimes we can be morons, that we think that more is better, that a little bit's good, we got a whole lot better. If a little bit of MS crystals is good in an application, why is a whole lot more better? Okay, I got a couple things. One, sucrose accumulation in a leaf is an important signaler for senescence, the die-off of a leaf. That if we get too much sugar for what the metabolism of that plant is functioning, it can signal that plant that it's time to start shutting down. Okay? That's one thing that I get kind of concerned about. Also, cytokinins is really important for regulating sucrose accumulation. Now, sucrose is, the MS crystals is like 100% sucrose, okay? Because it's sugar cane, pure, cold processed sugar cane that way, the MS crystals is. So, that's why we put on the program guides. We want to use about 1 pound of MS crystals per acre in an application, whether it's a foliar spray, a corn starter, residue spray. Now I didn't put in, I don't think I put in here on this, that bacteria. Some families of bacteria are more responsive to sugar sources than others. So to kind of keep the balance of our aerobic bacteria in the soils and on the plant, we don't want to overuse MS crystals. Now, I'm not going to say, has anyone used like high doses of MS crystals for insect problems, like in your garden or whatever? How much did you use?
Well, my wife mixed up about, 2 gallons in a sprayer and put it on like 6 grape plants.
Okay.
That was water, well, and I'm not even sure, I think she just used it like the MS crystal, like maybe a 1 ounce cup, and that makes a meal.
In there?
It was like 10 times more than what we put on a field or whatever. It
All
was
right.
successful.
What kind of root did you use?
It's probably enough to do the whole acre. I sprayed the raspberry plants. They had green worms on them.
Okay.
And the worms all, they left.
While I'm kind of bringing this up, it's kind of interesting. When we was at our dealer meeting, we was talking about the one dealer brought up the thing about the moron thing. It was interesting that when we got to talking about this later, he was the one that sat there and, was it Sam said he put on his potatoes, like equivalent to like 10 pounds of the acre of MS crystals in one application. Okay? And he was the one that just said the joke 2 hours earlier about the moron thing. I'm like, Sam, what did you just? He said, well, I was talking from experience. And it's like, okay. But he had a real bad problem with California potato beetle in these potatoes. And he had, I forgot what it was, a couple acres of certified organic potatoes. He already had them sold, doesn't have a buyer that was going to take all of them. And so he had these potato beetles move in. It looked like he was going to lose all of them. And he was like, well, I'm just going to dump a bunch of sugar in there. He dumped a bunch of sugar in there with his water, and he sprayed them on there that evening and come out there the next day, and he said about all the potato beetles was laying on the ground dead. Now, I can't deny in saying that, even though I have that up here, I can't deny in saying that didn't work. You don't need to do that again, okay? But, and what I'm talking about here is repetitive use that way. Now, there is some situations when you get like a bad insect infestation like that, running a rate up to 7 or 10 pounds of the acre equivalent for a one-time application, I'm not going to say that it's not going to work, okay? But for plant health, plant balance, and plant stability, we don't need to do that on a regular basis. Okay? I want to bring that up that way. That I'm not going to say that it doesn't work, because obviously I mean, I've probably got a person, at least one person in every meeting I do that says, well, I did that and it worked. And it's like, well, we don't want to do that repetitively, okay? So, that makes sense on the MS crystals. I mean, if a little dab's good, a whole lot isn't necessarily always better, especially long term. I mean, if you're needing some energy, taking a little bit of caffeine is really good, but we don't need to ingest large amounts of caffeine on an hourly basis every day. I mean, that's just not good to do that way. So, I wanted to bring that up about the MS crystals, okay? Now, does anyone, I know that was a lot of information on the hormone thing, but in relationship to like the foliar supplement and the kelp growth, and on using those and in timings on that, does anyone have any questions on something like that or had something happen? I want to address that.
So on corn, on silage corn, would you, I think it was earlier that you had said you would not recommend the kelp grow like in that 28 to 35 day period?
Correct.
Not at all?
I wouldn't want any kelp grow in that application. No kelp growth. I want it all cytokinin based because where it is, where we're talking about this resistance and also the thing on these proteins and sugars, whenever that cell division thing that is talked about there earlier, whenever that tassel is trying to form at that like 34 to 38 days after emergence, that tassel is trying to form and divide, cell division. All I want is cytokinins at that point. I don't really want to add any extra auxins. Now the plant's going to have some auxins there, but I would just leave them at their natural levels and their natural states. Okay? Now, if I know that you're doing silage corn and you're needing tonnage, we might want to use an equal rate of kelp grow to foliar supplement in the starter. Okay? To try to help get that plant up to size quicker. Okay? Or to have more size, because remember, that tassel forms at a day length thing from when the photosynthesis time starts. So, I've seen 32-day corn this tall, and I've had 32-day corn I've foliar sprayed and I break it off with a tractor trying to spray it. I mean, I've had 32-day corn this tall, okay? A lot of that has to do with how quick it comes out of the ground and a lot of other factors that way. But putting some kelp grow in a starter would be fine. The 32-day spray, the foliar supplement only, no kelp grow, okay? I can't emphasize that enough. Now, if you want to go back to kelp grow later, let's say up 40 or 50 days after emergence, you want to come in and do another foliar spray. If you're going to do it with silage and you want to throw 16 ounces of kelp grow in there with foliar supplement, that's fine. Yes?
Now for alfalfa, would you say that'd be recommended after every crop or like in between every crop?
Yes. And that's what I would prefer to see is like after each cutting, come in with a foliar spray, like 50% foliar supplement and whatever rate of kelp grow over that, like 10 ounces and 20 ounces or 15 ounces and 30 ounces or however you want to do that way. Comparing foliar supplement to kelp growth. Because we're wanting to keep that primarily in the vegetative stage that way.
When you spray that, then what you're just talking, I mean, is that as soon as you take the crop off or wait till it gets up so high?
That's going to determine a lot by temperature and soil moisture. What I tell everybody is I want enough regrowth to where all of that spray ends up on plant leaves, not necessarily going to the ground. It's not, if it goes to the ground, it isn't like it's totally lost. But, as you've seen with this hormone thing, if I can get it on the leaf and get it to absorb straight through the leaf, I can make bigger changes and quicker changes than I can going through the soil and back around. Because the soil and back around is going to be more delayed. We see that a lot, especially with produce. Running kelp grow through the drip line, you're going to get a more longer curve of growth than it is if you put kelp grow in a foliar spray. You're going to make a quick change right there. So the big thing is having enough plant growth. Now, obviously, if you've got a grass legume mix, you're not going to have near as much bare ground in between plants as if you've got a straight alfalfa stand to where you go in and cut it and it's like, okay, I got all these little sticks sticking up. We need to wait till it's leafed out enough to where we about got 100% ground cover. Okay, does that make sense? Because I want it all to go directly in the plant. Brian?
Yeah, I tend to see the tendency that fellows want to wait till they see a lot of growth there. And I think it's better if you get it earlier, again, still so you get it all in the plant. Because that plant is going to have, you know, more days of growth to be able to respond to it. Because sometimes the guy's waiting and he can't get up there when all else is tall. And I think we minimize the response that we could have gotten if we would have gotten it down earlier. So I lean to earlier versus.
Do you want to do that before rain yet, or is that, you want to just leave that on the leaves then?
Ideally, leave it on leaves for
Right.
2-3 hours at least.
Yeah, you want to soak in that way, not necessarily be washed off that way.
Oh, that one guy I was talking to said evening is the best time to spray on that type of stuff. It uses metabolizing it the best.
For metabolism and also for the plant to be able to absorb it too. Especially if you're really warm on temperatures. The pores on a plant is just the opposite of what the pores in our skin. When it's really hot outside, our pores open up for us to sweat. Plants are just the opposite. When it gets real hot, their pores close up to retain fluid and keep them dehydrated, to where you get in the afternoon, once the plant senses that the temperatures are starting to drop, it's preparing for that point of what meteorology people calls the dew point, as far as where the water level in the air and the temperature meet. That's technically right before then is the ideal time to do a foliar spray. Because when you get to that point, the plant's anticipating the dew, it knows the temperature's dropping, and all the pores open up, and that way when you put a foliar solution on there, it's going to absorb it really quick and metabolize it and go to work. So, now, and that's not to say that if you have a cold front come through, let's say in the middle of the day in the summertime, to where you have that meeting effect, the dew point temperature is met, let's say at 1 or 2 o'clock in the afternoon. It's not to say that at that point you might be able to go out and foliar spray right then, because the plant's done met those requirements and it's opened up. Does that make sense? It isn't just necessarily, oh, I got to do it at 8 o'clock at night. That if you get a temperature or weather system come in even in the middle of the summer, or a thunderstorm late in the afternoon to where it cools things down and it don't ever really rebound in temperatures, you might go out and foliar spray right after that rain that way. If you didn't get too much that you're going to mess up your hay stand or whatever. So, does that make sense? Yes?
Where does kelp grow come in for grasses to head out to, like in pasture?
On what now?
Like kelp, like pastures, like grasses that would head out early?
Okay.
Where would kelp would come into that?
The kelp, the auxins is definitely going to help where you get some plants want to head out early, like I was talking about on the alfalfa, anything that's wanting to mature and go to maturity early. The auxins is going to keep that plant in that vegetative state and keep it going. Now, like Brian was saying a while ago, if we go in and cut it or graze it, we come in and do some grazing that way, we stress that plant. Remember what I said there earlier about how cytokinins helps a plant in recovering from stress and be able to making switches that way? That's another reason why, like Brian was saying, I want the ground cover to catch all the spray, but yet I would like to catch the plant as young as possible to help it cope with that stress and try to get it rebounded on the growth side as quick as possible. But if you're consistently having a field or particular plant that's wanting to go into reproduction too quick, you may want to look at increasing the kelp grow rate in your mix to try to, that to me would be an indicator that that species or that particular situation, the auxin levels is getting too low in that plant consistently, and then we need to try to pull that up even higher. You may want to bump that rate up on the kelp grow that way. So, anybody else on this plant hormone thing? I want to make sure that I address that. So, I don't know, I was talking there earlier about some of the changes and things that's happening. I'm kind of excited if this kelp thing, I mean, the way it looks right now, if this kelp thing kind of works out, that hopefully this thing on this foliar supplement, or Aqua 10, as we're, and I didn't really talk about that. That Aqua 10 name, there we go. The gentleman that taught me on how to do kelp extracts, that had actually been working with kelp extracts and harvesting kelps since back, well, about the time I was born. He started back in 1971. On that, his laboratory that he had set up for doing all that, he called it Aqua 10 Laboratories. So that was the reason he passed away here about 10 years ago now. So when I was looking at the name change, and actually my dad and I was looking at this name change back, I guess that was like in 2018, 17 or 18, we was looking at possibly going to this. Matter of fact, I already had this prototype for this label made up then. And my dad didn't like change. Name changes or even phone numbers. I mean, if you was going to change your phone number, I mean, he would just have it fit that way. But, so it kind of got shelved. And since I'm going through some registration things in some different states, I thought, well, now's the time to make a name change. Particularly one of them was California, so they've got a lot of hoops that you have to jump through out there. That that's not the reason why I've had some people say, where do you get this Aqua 10 thing? So it's kind of, I guess you'd say, kind of a memorial for Bill Campbell that way on what the research and work that he done with kelps for almost 50 years that way. So that's kind of where that name thing comes from. So, any other questions on the hormone thing? No. Okay. I got Brian, you was wanting to go through a thing on the starters, wasn't it? Or do you think what we've done covered that on, what you want to cover on the starters?
I don't know, otherwise if people want to just have this an open discussion on whatever, if anybody has questions on anything, you can maybe go that route too.
This on, now there earlier, that's a picture of a couple calves from my hand. Okay, now, we talked about earlier, a good point got brought up on the thing on mixing the kelp growth. Has anyone got any other type of mixing dilemmas that you've had with the zone product stuff?
Don't mix liquid cal and any, or I don't remember what that stuff was, it turns to jello.
Well, like I was talking about, the pH difference between the liquid cal and the kelp grow, also the liquid cal and the soil supplement will do the same thing. Okay, the soil supplement will have a pH of 8.5. Okay, it's a little higher pH than even what the kelp grow is. Okay, TR, like I mentioned earlier, TR3, BR5, for conventional guys, the liquid cal, all three of those are very acidic. And then when you just try to mix something acidic and something that's higher pH, you're going to have a conflict. okay?
It only takes a little bit, about this much in the bottom, Brian, we figured, oh, there's such a little bit in there, it ain't gonna matter. But it did.
Well, and I've had people do that, it's like, well, there's just a little bit in there, I'm gonna go ahead and throw some 318 on top of that, and it'll do the same thing too. But that, so, anybody yet? Yes?
My question was, on a liquid soluble K, you don't have something together on that, do you?
To liquefy it better?
Yeah.
No, that's just kind of
That's just a,
the nature of that beast, I guess is the best way to say it. Now, I will say this, with the organic or soluble K, the soluble 0052, all those same thing. The hotter the water is, and actually in that situation, the more acidic the water is, the better it's going to go into solution, and the quicker it's going to go into solution. Okay? So any of you are dealing with the powdered potassium sulfate. The hotter the water and the more acidic. So if you're going to be, say you're making a mix and you're going to have some TR3 and DR5 in that tank mix, it ain't going to hurt to put all the TR3 and DR5 for that whole tank mix in a bucket with hot water and then try liquefying that organic. That's going to be your best possibility. That stuff is just hard to get mixed. And if you can give it a little bit of time too. If you know that you're going to be using some of that, of trying to give it a little bit of time. I mean, if you're like me, a lot of times I'm mixing up foliar mixes and it's like, man, I should have had this done an hour ago, and you're trying to throw stuff together, and yeah. That's kind of the best solution on that.
if you're using it in a starter, 2 pounds per acre is about max. Corn starter.
So you're using fish?
Yeah.
You are. That will be to your advantage over like something like the 31818 or 9189. Because those type of fertilizers just neutral pH, where your fish has got a pH of 4. That acidity, I mean, if you can do all you can with your water and TR3 and BR5 to try to get it dissolved, you get most of it. Then if you go and you put it in your fish and you can agitate it at that point with the fish, most of that should go ahead and dissolve, because that other additional acidity of the fish should help with that.
For seed germ though, you can overdo that, right?
So that's pushing it.
2 pounds.
2 pounds, that's pushing it. Yeah, that's pushing it. Even though there's sulfur there, that product still has a certain amount of salt index that can work on germination. I wouldn't want to do that in sandier soil. Higher CEC soil, you can get by with more of that than you can. And I'm saying higher, I'm talking about like 15 and above. You start getting CECs at like 6 and 7 and below. I don't know if I wouldn't recommend 2 pounds. Just because of that potential there.
Do your products have mollies and cobalt?
The VR5 and TR3 has a little bit of molybdenum in it. 0.02% molybdenum in the VR5 and TR3. There's no cobalt in either one of those two. Now the macronate product has cobalt, selenium, and iodine in it. So you can get your cobalt from there.
So, you have to show deficiency.
As far as applying the macronate to the soil, I really recommend applying those type of nutrients and the macronate in foliar sprays or your starters, other than just a general broadcast. It's not to say that you're more or less wasting your money, but you're not going to see as much response in plants and plant metabolism just broadcasted over kind of isolated that way. I think that kind of like this energy thing that Brian just talked about a while ago, in agriculture, in the United States, modern agriculture, we have neglected some of these micronutrients like cobalt, selenium, iodine, and molybdenum. We've not been really keeping up with those like we should be. And I think that's one of the things with some of these energy level and things like what Brian's talking about. I think our traditional NPK usage and just repetitively using that we have neglected some of these micronutrients, and that's what some of our energy numbers is kind of. And it's like I've heard Brian talk about with forage samples. You take a forage sample, I mean, this should feed out good. Get cows, and milk production drops off. You get another one, man, this one don't look too good by traditional testing. Give the cows and milk production goes up, then it's like, okay, there's another factor that's not showing up on the paper that the cows are responding to, okay? And sometimes I'm going by, now I'm getting to where I'm looking at, okay, where is the cobalt level, okay? Because cobalt, has anyone ever heard or went and got a B12 shot? Heard of someone going and getting a vitamin B12 shot and it give them energy? It used to be, that was a big thing. I remember hearing people 30 and 40 years ago going and getting B12 shots to give them more energy. B12, the primary element used to make B12 is cobalt. So it's like, well, why isn't that promoted like it was before? But another hand, yes.
You're talking about the MS crystals being, so you use that for a sugar source. So more on the animal side here, what's the difference between the MS crystals, say, what's the benefit, say, in using MS crystals versus like molasses?
In any application, you got something particular you're thinking about?
Animal. My situation would be very
Okay. With biological life, I'm going to kind of start there, like with your infix, molasses, as far as I know, I've done any checking here in the last couple of years, but all of the molasses in the United States is being imported in the country, okay? Even if you buy a food grade molasses, which should be a grade for us to be able to eat, the food grade laws only apply to the molasses once it gets to the United States. So before it gets to here, they can technically treat it and do whatever they want to it. And as long as it gets to here, they start treating it as food grade, it's food grade now, okay? So, now this has been going on for a long time. It's the reason we started with molasses. If you go back and you get an old yellow book, one of them that was printed from back in the 80s, it's had in there using like a quarter molasses per acre in foliar sprays and starters and everything. We got to running into issues with the more work we got to doing with bacteria, we were getting inconsistent numbers on how they were multiplying and living. Well, we got to do investigating, and the molasses had growth suppressants added to it to keep it from wanting to grow mold or other bacteria growths in it, because it's a liquid, it's sugar. I don't know if you've ever made like your own maple syrup, and if you minimum cook it, and if you don't seal it and you let it set out in open room temperature, what you start getting on the surface? You get a little bit of mold growing on the surface, right? And you skim it off. I mean, the syrup underneath is still good, and you skim that off. Well, to keep molasses from doing that, they add formaldehyde to the molasses, and that way it don't grow them. But formaldehyde will kill any of the bacteria in the infix, or even earthworms and other stuff that's already in your soil. So, now, it's kind of what Brian had up here on the energy thing. there is sometimes things that can produce a lot of energy, chlorine, fluorine, and bromine, that's kind of detrimental to life, okay? So even though formaldehyde would not be something to be directly detrimental to a plant, I think it would be indirectly, if that makes sense. That's the reason why we kind of shied away from, to give you a little bit of history on sugars, we started out with food grade molasses, then we went to high fructose corn sugar, okay? We made the switch out of the molasses, that would have been about 1986, 87. And we got started with high fructose corn sugar, cold processed corn sugar. We used that until about 94. Well, 94 is when GMO corn started being sold on the market. And the group that we was getting the high fructose corn sugar wouldn't guarantee us that they were going to use all non-GMO corn. So then we had to kind of back up and rethink the whole thing. And that's where we end up going to, at that point there in 94, 95, we went to a cold processed cane sugar that way. And I hope it's going to stay non-GMO. I heard that last year the country of Brazil has come out with a genetic modified sugar cane. So I'm hoping it don't get out of the country of Brazil at this point. So that gives you a little bit of background on sugars and sugar sources and why I'm not really a big, now, as far as now I've got customers like down in Tennessee and Arkansas and places where they make their own sorghum molasses, where they're raising their own sorghum, they're pressing it, they're cooking it, they know they're not I'm like, hey, if you want to use a quart per acre instead of the MS crystals, that's fine. Up here, now, most people is not going to take maple syrup and use it to spray on their cornfield. You can get more money selling it than you can spraying it on your cornfield. But, and some of those guys down there is that way too with their sorghum, that they can get big money down south out of good quality, light colored grain sorghum. Man, it brings big money. But they have to get a batch that turns dark on them. They'll use it on their crops because no one will buy it that way then. So if you're raising your own honey or your own maple syrup or whatever, where you know it has no preservatives in it, you want to use it as a substitute to the MS crystals, that's fine. But the preservatives is what's going to cause the factor that way. So.
At what rate would you use like maple syrup or something like that? Versus
depending on kind of how condensed you're making it, I would probably, you could go back to, if you get it close to what a high bricks maple or high bricks molasses is, which that's, let's see, what is that, like 85 bricks. If you get your maple syrup to that point, I would say you should be able to use about that same rate, 3/4 of a quart to a quart per acre. Now, if you have to get some that's a lot lower bricks, you can just kind of adjust it accordingly that way. I seen another hand.
This is kind of off on something different, but the macronate, is that one approved for organic? I know it's not OMRI listed. Are you working on that, or?
I'm not working on the OMRI listing part of it yet. I think we got through all the hurdles and everything with a lot of the certifiers. I don't know who you certified with.
NICS.
You're with NICS, okay. I don't know if I ever talked to anybody this past year from NICS on that. OFA from Ohio I have, OCIA I have. And we got that all cleared with them. That was kind of an ordeal. They had one ingredient they said, oh, you could use for plants, but the other ingredient could be used for organic animals, but you couldn't use it on organic plants. And don't ask me how they come up with that. So it's like, if you could use it for one organic, you can't use it for the other organic, why can't you flip the I went through a bunch of hurdles with them on that ordeal. So, but I think at this point, those certifiers I have. You may check back with Nick's. I don't remember, because my secretary, she was kind of, once we got that packet cut together and the letter and everything in there, she was started just sending them to those that was calling, certifiers calling in and asking, so I don't know. It may be fixed with them. If not, ask them, and if it isn't, then tell them to get a hold of us. And I can get them that paperwork, and hopefully we can get that taken care of. Yeah. Yep.
Talking about that NICS, using the Bore Extra, they require deficiency testing.
And that, well, any trace mineral is going to be that way. Technically, if you're going to use the TR3, you're going to have to show manganese, you're going to have to show the deficiencies, and for the micronutrients, you're going to have to show a deficiency too. I wouldn't suggest, if you're going to do testing for those, I wouldn't really suggest using a soil test, because there's not as much research and data where the optimum levels should be for like cobalt, selenium, and iodine in a soil sample. There is with plant tissues and forage test or something like that. I would rely, they're going to, how would I say, your certifier is going to come near taking it and accepting it if it's more from a plant type test than it is a soil test. Does that make sense? I've just found that in my experience of talking to the certifiers from the other end. I mean, they talk to you all, and then I get usually someone else in the group is the one that calls me, and so you know how it is when you pass some stuff around. Sometimes it gets mixed up, but that's what I recommend everybody to do. That if you're wanting to use something like that, now the certifiers I've talked to, a forage sample or a plant tissue sample, a deficiency of a trace mineral will work for that given season. They're not going to let it roll over from one season to the next. Okay? If that makes sense. Now, like zinc, copper, manganese, and boron, because where there's established levels on a soil test for them, most certifying agencies will let you go 2 or 3 years on a soil test on showing a deficiency to get to use it that way. Okay? Thanks for bringing that up. And I would say it this way, and I probably said this before in a meeting, just because your soil test is showing an adequate level on, let's say, zinc, it doesn't mean that you can't have a stress or something happen during the growing season and your corn plant or grass go deficient in zinc. Okay? If you suspicion that plant is low, pull your plant tissue, and if that plant tissue is showing a deficiency, that will supersede your soil test, if that makes sense. Okay? So don't just think, well, I can't ever use any TR3 the whole season. That isn't necessarily true. You may be able to pull a plant tissue and prove that you can use it that way. Okay? I see another hand a while ago. Or anybody else on something else?
I
Yeah.
had something different.
Oh, that's fine.
Up in this area last summer, July, August, it was very, very easy. And they blamed it on the Canadian wildfires. Of course, then everybody was all concerned about lung health and all that stuff. But then about a month ago, I was watching one of these talk farm shows, and certain areas had a good bump in soybean yields. And they credited it to the wildfires. It was giving more sulfur in the air and making the soybeans do a little better. I don't know if that was true or not.
Well, you should record that and send it to your U.S. representative and send it to try to get the sulfur back on the piece.
Well, exactly what they do, they outlawed fires.
And I would say it's probably more the sulfur aspect of it than anything else, because you're going to get a sulfur out of that plants are deficient in.
Yeah, but then on the same subject, listen to Dr. Wallach, anyway, Joe Wallach, he said that people's health have declined from the 20s and 30s on because we burn less wood. when the wood smoke was in the air, people were healthier. You know, that was his big issue.
And it's funny, Brian and I was talking about this last night, on programming and conditioning people mentally, that if you tell somebody, it's kind of like, I forgot his name now, the guy that was in charge of the propaganda machine for Adolf Hitler in the 1930s. His thing was that if you tell a lie to a group of people long enough, they'll believe it. Well, it was amazing in our area, which we didn't see hardly no hate from all the fires. But people was hearing this news thing about this, and it's amazing the number of people that I heard that, man, I've been having trouble breathing the last couple of weeks. I haven't seen any, but they were watching, but mentally they were being programmed that this is an issue. I'm going to have a lung problem. And it's like, well, come on. I'm like, but I don't know. I mean, we as human beings, I don't know. We're supposed to be getting smarter, but I think we're getting dumber. I don't know.
Well, see, years ago when people used to cut their own firewood, they stayed healthy because they were exercising.
Well, and that's like, I had a gentleman,
I used to use the buxel, I used to work for a guy.
well, that's like
That would work.
20 years ago, there was a gentleman I went to church with, and at that point, I think he was like 80. So, I mean, we're talking about, he was born like 1920. And he was talking about how when he was growing up, and his parents, that all his mom would make homemade biscuits and have it just full of lard fat and all this stuff. And now all these people, oh, we got to cut our fat out, it's going to kill you and everything. Well, he was over 80 years old, and he said his mom lived to be 100. And he's like, why is this fat so bad for you? I mean, we grew up eating all that, but it was part of an exercise thing too. I mean, they had to hand chop all their wood, they had to do all this stuff, to where most Americans now don't exert themselves to do anything. Well, they don't need all that extra fat and that energy that way too. So, kind of what you're saying, okay, the smoke may have been helping too, but the extra exercise of cutting the firewood to be able to make that smoke may have been a big factor in that too. So, Ryan, did you have your hand? Okay.
Do you remember the number of elements that corn had in back in the 20s that are no longer in there?
That's no longer in there?
It's no longer in corn now. I've heard number, I don't remember, but just on that thought. So we were getting, back then, they were getting nutrition and we didn't get no more.
Now, I do know this. I've got a feed book at home that was my dad's, and it's copyrighted 1940. And in the back of it, it's got a chart for different grades of different grains. Okay? They had number 1 corn, all the specs, number 2 corn, number 3 corn. Do you know what the specs now for number 1 corn is? It fit the column for number 3 corn in 1940. We got a problem. I mean, it's like, well, I got number 1 corn, it's like that number 3 in 1940. So going back to what you're saying, there is a nutritional thing and there is a energy thing that we're not getting out of grains now that 70, 80 years ago, the composition of these grains was totally different too. I mean, that's just, pardon?
We feed more, so it's the same.
Well, and that's the thing, you get Americans, we've got all this food, and we're eating all the time, but you realize that probably, last time I heard, close to 50% of the Americans would be diagnosed as anemic. If you go out in the store, I mean, we don't look very anemic, okay? But it's like, we're all of a sudden, but it goes back to what we're eating doesn't have near the nutritional value, so our body is like, I'm still hungry. Because it knows the nutrient levels we need, and you gotta keep eating and eating. Yeah.
Try to get that weak link that you're not getting from anything, and you're never gonna get it, be satisfied until you get it.
You're never gonna get nutrient satisfied that way. So, okay, any other questions on the product or mixing or anything like that? I wanna make sure that I got that covered that way. So, okay, well, I'm gonna be around here, for however long. I didn't say what time we actually gotta be out of the building, but, that way, I'll be around here. If you got any questions or whatever, let me know. And, you got anything else, Brian, that you wanna cover or do or whatever?
I think we got it covered.
So, okay.
Okay.
Well, if we want to we can kind of stand dismissed, and we can kind of, hope you all have a safe trip home and have a productive year that way. Appreciate it for everybody coming out.